Okay, the second application we want to focus on today is the Rubicon Genomics Plasma-seq, ThruPlex Plasma-seq kit. And we've also automated that on the Biomek FXP hybrid system with the SPAN8 arm and multi-channel pod. So the ThruPlex kit is designed around processing cell-free DNA, cfDNA, of very low input. So what is cfDNA or cell-free DNA? It's highly fragmented DNA in the blood, and a high abundance of cfDNA in cancer patients is shed into the blood from tumor cells. So similar to the exosomes that Zach talked about, these tumor cells during necrosis are releasing fragmented DNA into the plasma, into the blood, and then plasma can be collected and analysis performed on those samples. So why is it important? The plasma, along with other liquid biopsies, is more easily available, easier to obtain, less invasive, as compared to solid tissue, solid tumor biopsies. And it's used in translational research in oncology, and research in that field can be conducted on cfDNA. And then there can also be research performed on using the fetal fraction obtained from maternal plasma in non-invasive prenatal testing, or NIPT. So this is the workflow of the Rubicon library prep system. Much simpler than what we saw with the RNA access kit, but still presents challenges from manual processing. So it generates NGS libraries from cell-free DNA isolated from plasma. It's optimized for extremely low input of less than 1 nanogram, and we tested down to 0.1 nanograms of input all the way up to 30 nanograms. It's fast and simple. It has a 3-step process that's performed in a single tube, or in our case, a single plate on the automation deck. There's very low PCR duplicates, providing maximum library complexity, and I guess it does that twice. It's really good at it, and it's true. It also preserves the GC representation of the input DNA. So workflow challenges. Low input samples require assurance of optimal transfer, maximal sample recovery, and optimal pipetting precision. So the automation system is going to be able to handle that much better than performing these tasks manually. The repetitive pipetting of enzymes, indices during sample cleanup are also going to be prone to errors and fatigue. And then also the workflow is relatively simple and short, but there's 3 user interventions for adding sample to during the incubations and reactions. So you have to intervene with your method 3 times in 3 hours in order to get your work done on the thermocycler. So we've been able to integrate thermocycling capability to create a truly walk-away process. Okay, so the automated method features and key benefits are the ability to generate anywhere from 1 to 96 libraries in less than 3.5 hours, and that includes the final Ampere XP cleanup on the amplified product. So there's workflow flexibility built into our user interface that allows you to run either the library preparation portion of the method or the cleanup, or both. We have integrated T-Robot on-deck thermocycling for true walkaway capability, and built in, again, some index transfer options. Rubicon's kit comes as a 96, 48, or 12 sample kit, and they provide a 96 well-arrayed index plate, a 48 well-arrayed index plate, or 12 individual tubes. So we have the capability to do the pipetting from any of those labware types, either with a direct transfer A1 to A1, or with custom index mapping with our transfer by file feature. Again, optimized pipetting is going to deliver highly accurate and reproducible transfers of small reaction volumes, and that's going to ensure the most robustness of the chemistry and help maximize yields. So here's a slide of the Rubicon workflow, much simpler than the access protocol. There's 2 components that you'll be able to execute through the user interface. The first column is the full library prep protocol, and then the second column would represent the Ampere XP cleanup. The slide to the right, the table shows the processing time, and you can see that there's minimal impact of sample number on the time that it takes to process. So relatively similar time frames for processing a low throughput run as well as a high throughput run. So again, all the methods have our user interface and reagent calculator. Just to reiterate a couple of the key features of those. The user interface creates flexible workflow options, allows you to choose your index transfer option. It has a trusted Ampere XP process built into it, which is not part of the Rubicon method, although we've added that as an extra feature. And then optionally, you can run a T-Robot integration to do on-deck thermocycling and create a walk-away system. And again, the reagent calculator is going to be presenting master mix information and specific reagent volumes per well, and instructing the user where to place that on the deck to minimize error. Okay, so these slides represent the library yields and a bioanalyzer tracing to show the consistency across the input range. So we have 0.1 nanograms up to 5 nanograms in those top 3 slides, and then an NTC non-template control on the bottom right. So each of those inputs requires different thermocycling cycle conditions. So after all is said and done, and each of them are differentially amplified with their respective cycle number, you can see the consistency between a low input sample, 0.1, and a higher input sample of 5 nanograms, with median sample yields of approximately 25 to 35 nanomolar, regardless of the sample input. Okay, so folks at Rubicon took care of the sequencing for us and handled the bioinformatics. And just in summary, the sequencing data demonstrates highly accurate and reproducible automation protocol generating metrics of greater than 50 million reads per library with very low % duplication. So you can see from the different input types that the library size is very consistent. There's minimal impact on that in the process. And then % duplication is extremely low, less than 5% for samples at 1 nanogram, 5 nanograms, and we also have 10 nanograms on this slide, with slightly higher % duplicates for the low input 0.1 nanogram samples, but even those are less than 20% duplication. So with that low of input, those are extremely low PCR duplicate numbers. All right, in summary, the Rubicon kit is capable of generating 96 libraries in about 3.5 hours with low input cfDNA. Reduces errors from repetitive pipetting. It has truly walk-away capability with our T-Robot integration and highly reproducible results with low PCR duplicates from all sample inputs, including the 0.1 lowest end. And 0.1 is actually below the range they recommend for their kit, but we took it down that far as to push the limits of the system, and it performed very well.